بسم الله الرحمن الرحيم. السلام عليكم. أنا يسري عبد الله طه. Um, um, I'm delighted to present the grand round this month on the chosen subject of antivirus, non-HIV antivirus, and for that matter, maybe also non-viral hepatitis antivirals. And I said, my name is Yusri Taha. I'm an infectious disease physician and a virologist. I graduated from the University of Khartoum, or in the University of Khartoum, and trained in initially in Sudan as a house officer, and then in the UK from 1997 until recently. Been working there as an infectious disease physician and a virologist. My interest has been in within viral disease in herpes viruses and viral hepatitis, and I'm working now in Saudi Arabia in a sabbatical leave for some time, and the hospital I work here in Saudi Arabia is called King Faisal Specialist Hospital and Research Center. So I'm enjoying Saudi Arabia now. It's quite busy hospital, lot of infection, and because it's mainly a transplant center. So that's really me, and I think we should just crack on and start. The headlines of the book, maybe before that, you probably noticed from the title that we're not going to have a traditional lecture on antivirals. I thought rather I would just choose some topics from across the field and discuss with you today, and use the topics that I chose to highlight the basics and the principles and practice of antivirals, rather than going anything like traditional. So two parts of the headlines. The first part, 1 and 2, the antiviral landscape before COVID hit us, and how we classify or group antivirals, including how they work. And in the second part, and I hope we find time to do that, I thought probably better to zoom into a group of viruses that we kind of as clinicians are busy treating with HIV out of the way. I could have chosen either viral hepatitis or respiratory viruses or herpes viruses to do what I wanted to do. So ultimately, I decided to do herpes viruses, and I did my doctorate, by the way, in varicella zoster virus molecular epidemiology. So it's a subject that's close to my heart anyway. And as I said, if we find time, then we choose something emerging rather than the herpes virus that has been with us for millennia, to see what lessons we've learned in recent years on antivirus, especially Mpox and COVID-19. Okay, and please, if you want to ask any question, I think just interrupt me, and then we can discuss things further, because I would like it to be more interactive. As I said, it's not just the disease that we deal with every day in hospital that require or need to have more effective antivirals or safe antivirals. In fact, developing effective and available and kind of readily available antivirus agents is a global health priority. That is very well described, and maybe the COVID-19 is the best example. And several global trends will make pandemics more likely to occur in the future. Climate change, loss of habitat and fragmentation, destruction of wildlife, serve to increase the animal human interaction and the risk of zoonotic spillovers. And also the warming temperatures increase the geographic area of where viral vectors like mosquitoes and ticks find it more hospitable to live, potentially increasing the risk of spread of arboviruses. This map here in the middle shows the current worldwide distribution of the mosquito Aedes aegypti, the mosquito, the vector for yellow fever and chikungunya and Zika and dengue fever. And you can see here that the duration across the 12 months of the year in red, the red is 12 months, though, so you will find the mosquito flying around for 12 months a year. And also the distribution around the world. You will expect, if the temperatures carry on like this or worsen, both the duration of activity of mosquitoes and the area of spread is going to be bigger. The third thing that is, or fourth thing that is very important in the risks of having pandemics is the global travel. Global travel, the prevalence of it is just very high. It dipped during the pandemic, but in 2024, this year, the size of travel between countries will surpass the levels that we had before 2019. And this type of trouble can easily change a local outbreak into a pandemic. So we need antivirals, so we need to be ready for that kind of thing. So first, the landscape of antiviral prior to COVID-19. COVID-19 is being used as a watershed, like a landmark to kind of describe a lot of things, anything that happened before COVID and everything that happened after COVID. Before COVID, this graph, I like it so much. It's been, I think, published in 2016 by de Klerk and Lee. And you could see here that in 2016, and by the way, between 2016 and the start of the pandemic, not many changes have happened during these 2-3 years in terms of antivirals. I will mention some of them later. But you could see here that the viruses to which we have antivirals approved by the FDA were around 9 viruses. And the medications, around 90 medications altogether. Mostly they are HIV. They have to have a different scale for HIV there to fit in. But there are other medications, of course, especially hepatitis C and herpes viruses and a little bit of flu, both Hep C and Hep B. The graph also showed the date when the first antiviral was approved, 1963. Hydroxyuridine for herpes. Same year is a year where hepatitis B was discovered. Human papillomavirus was discovered 2 years later, and then, of course, in 83, HIV, in 89, hepatitis C. These are the viruses to which we have got approved antivirals. The other 5 or 4 viruses were known prior to 1963. You have to remind me later to talk to you, if we find time, about this year, this circle, where everything seemed to have taken off, mid-90s to around 2005. Loads of things happened that probably require a lecture on its own. But I will mention the end of the 70s, 1980, when acyclovir was approved. Acyclovir has got a special place in the history of antivirals. It's the first successful antivirus and still going strong. We use it every day. It is a guanosine analog, but it is acyclic. So it actually used to be called guanosine or acyclic guanosine rather than acyclovir, and is extracted like many nucleosides from natural sources, like this fungus from the Caribbean. It's called C. Crypta. The work and the way acyclovir was developed took a decade or so, but these two guys, Gertrude Elion from New York and George Hitchens from the Wellcome Lab in Kent in England, among others, really worked on this. And Elion in particular is the person who was credited with describing how acyclovir works on specifically on herpes simplex, because he described that there is a virus or a virally encoded protein called the HSV thymidine kinase. It phosphorylates the prodrug, the acyclovir, into acyclovir monophosphate before handing it over to cellular kinases to make it diphosphate and triphosphate, and then the molecule will inhibit the HSV DNA polymerase, stopping replication of the virus, and can also insert itself as a monophosphate into the growing chain and just terminate the chain in a way similar to what we see in NRTIs, for example, in HIV. These two guys, with some of their colleagues, actually were awarded Nobel Prize in Medicine for this work and some other related work. And of course, it was this thinking of rational design of drug that kind of paved the way to a new era that gave us the antivirus, most of the antivirus that we have now. So let's go very quickly, to describe the 4 or the 9 viruses that we just spoke about. In 2016, we had antivirus against 3 DNA families: the Hypnoviridis, Papillomaviridis, and the Herpes viridae. And these families have got corresponding 5 members. So herpes viridae have got 3 members that have got medications, and you could see here there's a notable absence of EBV, for example. EBV doesn't have approved antivirals. We've got no approved antivirals for HHV 6 or 8. Still better, they've got 3 members, but the others do not. The virus is so different. They're different in their sizes, in the size of genomes, in the way they look, and also very important to antivirus and where they are actually in nature. Do they have reservoirs? Do they affect animals? We think hepatitis B have got a reservoir in nature, possibly bats, but that is not very clear. Human papillomavirus is actually also found in many animals, but there's no clear transmission between human and these animals like birds or reptiles and other mammals. And of course, the herpes viruses are our viruses. These are been with us for millennia, and they have got no animal reservoir. So since then, since 2016 or 2019, say if we speak about COVID, there's a new family of DNA viruses. It's also double-stranded DNA virus family that has got an approved antiviral. Can anybody guess which new family of double-stranded DNA virus that have got an approved drug? There are ones that are missing. Adenovirus is missing there, very important. But we have got now the poxviridae joining the club, with mpox as a member and smallpox, of course, having approved antivirals. Okay, the pox viruses, including mpox, is the reservoir for it in nature is rodents. RNA viruses, on the other hand, are they have got mostly got reservoirs in nature. We know about the HIV situation and the how they came from HIV-1 from chimps and gorillas, and the HIV-2 from cytomegalovirus. We don't know about hepatitis C. We think it's got a reservoir, but we have no idea where it is. The flu, the orthomyxovirus, of course, and the viral genomes are single-stranded in the all families that have got antivirus against them. Some of them are negative sense, some of them are positive sense, and that makes a difference later on when we have the next slide or two, where the antivirus will work. Do they work in the cytoplasm? Do they work in the nucleus? So anyway, since that time again, we have got a new family that joined the flavi and the paramyxos and orthomyxo and retroviridae, which is the coronavirus, of course, with SARS-CoV-2 now joining the ranks, and we've got medications that are approved for it. SARS-CoV is quite large, 30 kilobases compared to the smaller ones otherwise. So that's very quickly what we have. and what we have now in terms of families that have got approved antivirus. Now, I have to apologize for this hideously looking, schematic, but I'm not being inconsiderate with a haja. I'm just maybe a bit ambitious. So because I think if we can go through this, and if you bear with me, I think we will be done with the lecture very well. So this is a life cycle of any virus at all, and possibly where it can be kind of targeted in way of antivirus. So can I start? Okay, so we've got a virus particle trying to access a cell, or a cell trying to usurp or take in a virus, thinking that is food, thinking that is a nucleic acid or nutrients. So the virus get attached through a receptor, and then get fused with the membrane of the cell to release its genetic content in the cytoplasm, or it get endocytosed by the cell, and then inside the cell it gets uncoated, and with the same resultant outcome, the nucleic acid of the virus is released into the cytoplasm. Then from there, it depends on the nature of that nucleic acid, if it is an RNA, or if it's a retrovirus RNA, or if it's a DNA virus. So the RNA that can replicate in the cytoplasm as positives, like hepatitis C, for example, will be replicated with the aid of the viral polymerase. Things like the NS5A, NS5B proteins, to make new genomes, either mRNA to be translated into new bits and pieces of the virus, or genomic nucleic acid that can be used to make new variants. That is one way of, if it is an RNA virus that needs to replicate within the cell, it will just go this way. If you follow the arrow, it goes inside here, and there is a viral RNA synthesis that will take place here, and the viral mRNA and the viral genomic RNA will be transferred back out of the nucleus into the endoplasmic reticulum to make new bits and pieces, for the viral genome to be packaged into a new variant, and that new variant will be released from the cell, either by just budding out or by exocytosis, in a way analogous to the way they came in. If it's a retrovirus, on the other hand, the retrovirus will have to be reverse transcribed first, of course, using reverse transcriptase enzyme, and then from there, a provirus or a DNA will be formed, and that DNA will be transferred across the nuclear membrane into the nucleus, where the integrase, the transfer and integration is done by a certain enzyme. Then it becomes integrated in the host genome, and from there it gets translated or transcribed like any other DNA gene, to form new mRNA that can be translated into new bits and pieces, and also genomic RNA that is going to make new variants. DNA viruses, like any DNA virus, they come straight away into the nucleus, and where they get replication, transcription of the DNA, with the influence of the DNA polymerases here, and the same thing happened to them. Once the DNA is made, the viral DNA is made, it gets transferred outside, and it can be packaged with the translated other proteins to make a new variant that will leave the cell. Okay, so that will happen. Is just what the difference is, where it happens and how it happens and what kind of enzymes are involved. This is the general thing. Of course, each virus will have a little bit different from the other, but this is the general schematic. The red bits and pieces there are the places or the areas where they are currently, we've got antivirus that approved to target. For example, here, this 8 here, this is an entry inhibitor, like the entry inhibitors that we know for HIV or COVID, for example. the viral encoding amantadine for flu, the reverse transcription inhibitors are NRTIs and NRTIs, the polymerase inhibitors like the sofosbuvirs of hepatitis C, or the RNA polymerase inhibitors inside the nucleus, like favipiravir of flu, or the DNA, of course, synthesis blockers within the nucleus. And these, of course, are ganciclovirs and acyclovirs and foscarnets and the, of course, the integrase antivirals, integrase inhibitors, and so on. So these are the types of antivirals. And this chap was his name, de Klerk and Lee, divided them into 12 groups. But it's not completely kind of logical, because you could see that here they are mentioning NRTIs, protease inhibitors, and then they say influenza virus inhibitors. But influenza virus inhibitors have got some that are polymerases, and some are encoding ones, and some are polymerase inhibitors, while hepatitis C, NS5A, NS5B are polymerase inhibitors. So while here they're mentioning the enzyme that inhibit a step, here they're mentioning a name of a virus specifically, just to fit the narrative that they were presenting. But nevertheless, it's useful. So I'm going to do a very quick exercise. I mean, if anyone want to open his microphone and chip in, I'll be happy. So I've got here numbers in red, and I've got here letters in blue, and I've got examples here with each letter. So I want to arrange this. So A, lenacapavir, favipiravir, and zanamivir. To which group would these three antivirals belong?
12.
So 12, absolutely. So who's that?
That's
I can't see
Medusa.
that. I can't see
I think that's the
who's
only
that?
one I know. Khalid.
Come on. So B, glycopavir, graciprevir, and pretiprevir. So these are hepatitis C
6.
antiviral, صح?
11. Yeah.
But they're not 11. These are
I think
protease
6.
inhibitors. That's one of the problems with this classification. These are protease inhibitors, and the when you see pre or the PR in the middle of the word, like previr, pretiprevir, graciprevir, you know this is a hepatitis C protease inhibitor. So so this is actually class 6 or group 6 in this classification. Okay, while the dipasphere, sofosbuvir, and filpasphere are actually the NS5A and the NS5B. 11. And if you're not very familiar with the hepatitis C medications, if anybody says to you anything other than the previrs are all protease inhibitors. Everything else is NS5A inhibitor. Sofosbuvir is the only medication that is licensed on its own as an NS5B. It's the only one. It's like almost an orphan drug in that middle of one. There's another small molecule that is used in a combination, but this is the only standalone antiviral approved. Is the NS5B, is sofosbuvir. Okay, so what about D? Cidofovir, tenofovir,
6.
and adefovir.
6.
Number D.
6.
What do you think? So these are acyclic nucleotide phosphonate analogs, and they have got a different. They because that's confusing. They're called nucleotides as well. But as you could see, we use tenofovir for HIV and Hep B. We use adofovir for Hep B specifically, and sofosbuvir is mainly licensed for CMV. So they are a group, but they act in different viruses of different types. Okay, next, E.
8.
E. 8. Absolutely. So Marovrog, Stenofovir. So these are entry inhibitors and the RSV palifizumab. Foscarnet,
Scarlet should
Foscarnet is
be
also Foscarnet is a orphan. It's got no brothers and sisters. It's the
9.
pyrophosphate analog. It's 3. It's the pyrophosphate analog. It's got no
Okay.
molecule from that works the same way it does. Okay, what about acyclovir and famcyclovir and gancyclovir and valganciclovir and pamcyclovir? What about them?
9.
We've mentioned in the slide with Elian, Gertrude Elian and Dr. Hitchings. These are guanosine
Analogs.
analog.
9. Yes.
Yeah. Okay.
I
Acyclic.
got that.
Guanosine, but they are acyclic. They have got no cycle, so they're guanosine analogs. And the valrepine, entecavir, telbivudine, they're going to be number 2, the nucleoside, not nucleotide, nucleoside analog. So, and that's the difference between entecavir and tenofovir, and you can use that as a comparator. So, if they are nucleotides, tenofovir, nucleosides are entecavir. Of course, we've got the integrase inhibitors, and the first one, the deoxyuridine analogs, and our NRTIs and NNRTIs of HIV. Okay, that is the difficult one. The easier one, which makes sense and is possibly future-proof, is to classify antivirus according to the way they work, full stop. And this way is first neutralizing antibodies. So if it is neutralizing antibodies, that is one class. And neutralizing antibodies, of course, can be derived from human, people who have recovered from an infection or something. So it's like a convalescent plasma. Or you can take their lymphocytes and, then clone their, antibody-producing genes in B cells in large quantities in a factory, like neutralizing antibodies, and bring them back to use. Or they can be produced in small animal lab animals, and then after production they get like changed chemically. They call them humanized antibodies, or in larger mammals, or indeed in yolk of eggs. So we've got no time to go through them, but monoclonal neutralizing antibodies is a class. Okay, or you actually, instead of making an antibody that would block the virus from docking into its host cell, try to mimic the host cell receptor. So you make an antibody that is a recombinant human receptor, because the viruses can always change. We have done sotrovimab, for example, for COVID, and within few months, the virus just changed beyond recognition, and the monoclonal was just had to be thrown away. But the coronavirus remains, retains its ability to attach itself to the ACE2 inhibitor, ACE2 receptor. So if you can make an ACE2 receptor soluble, loads of it, that will distract the virus, and then you will protect the host from the infection. And of course, that is a very good idea, because, as I said, the virus will never mutate to the extent that it fails to attach to its receptor. So if you've got that receptor made, then you will overcome the problem of mutations with entry. The third way of doing or making antivirus, and this is also coming very strong now in research, is the antiviral CRISPR-Cas systems. These are as you know, the immune systems of the bacteria. They use them to break down phages and nucleic acids and viruses that are invading them. So the Cas systems cut double-stranded DNA and single-stranded RNAs. So for maybe decades now, people thought, we should use this. And we have used it before in a different way, when we found the systems in fungi, Streptomyces, that kills bacteria to stop, or protect itself from bacteria. And we kind of use that to produce antibiotics. So this is a very similar way. If we can use the antiviral CRISPR-Cas systems or deploy them, then we can basically kill or break down viruses. Interferons, all of us know about interferons and how they're effective in many viral diseases, because they are actually antiviral natural products. Peptides, on the other hand, or antiviral peptides, these are also coming strong, and we've got few approved peptides now as antivirals. And the one before last is the antiviral nucleic acid polymers. Many polymers are have got antiviral activities, but the nucleic acid polymers, specifically like the si and siRNAs, are powerful, and there are some products that are advanced in trials. I thought a product for hepatitis C should have been approved a few years ago, but I think now with the success of the other agents and the viruses towards elimination, possibly is not going to come quick enough. And the last group, which is possibly the most important group, because most of the medication we mentioned earlier belong to it, is the antiviral small molecules, which just block the virus at different stages of replication. So let's just take this, because it's going to be the slide before last, before we move on to a different subject, is how these 7 classes work. So the neutralizing antibodies are entry inhibitors. In fact, the first 4, we've got very specific or very clear mechanism of action. So the neutralizing antibodies inhibit entry and may signal to the immune system to send cells to come to fight. The recombinant human receptor, same way. The CRISPR-Cas9, I said it cuts the double-stranded DNA and the single-stranded RNAs, and the interferons, we know it degrades viral nucleic acids and also signal to the uninfected cells around the place to get ready to fight the virus or to stop it from being infected. So that is very specific, very well described function or mode of action. But the antiviral peptides, the nucleic acid polymers, and the small molecule antivirals can target different stages in the replication. So, for example, look at this number 5, the antiviral peptides. They can block the attachment, like Mercludex, that has been approved a few years ago, for a couple of years ago, it's not a few years ago actually, for hepatitis B and Delta virus. But also Telaprevir, if you remember, that's one of the first directly acting antivirals that was produced against hepatitis C. It's a protease inhibitor, but it is a peptide. Look at the NAPs. The NAPs also can do different jobs. The small molecule antivirals, as I said, this is the biggest one, and I'm giving you examples that are relevant to the talk about how we are using this system to produce new medications. So the small molecule antivirals can target the virus at different stages of replication. And the examples are the polymerase inhibitors, the NRTIs, the integrase inhibitors. These are all small molecules. But if we just speak about the polymerase inhibitors, okay? The polymerase inhibitors can be direct or can be mutagenic. So the antiviral stop the polymerase from working, or it interferes with its work. The product becomes mutagenic, so it wouldn't progress to a fully functional, mature virion. So the inhibitors that work straight away are like the NRTIs for HIV, like 3TC or FTC, while remdesivir will belong to the same group. On the other hand, the mutagenic ones, the best example for it is ribavirin. It's a polymerase inhibitor. It introduces just mistakes in the product, and thereby there will be no production of a good survivable or surviving particle. Molnupiravir belongs to that group as well, while the protease inhibitors for HIV and hepatitis C and SARS-CoV all belong to the protease inhibitors, of course, and Molnupiravir belong to that group. So you could see here from this column that we've got sotrovimab, okay? We've got the Pac-Man, which did not progress very well, but it was because we are not sure yet about the CRISPR-R, but these are trials for COVID. We've got remdesivir, we've got molnupiravir, and we've got Paxlovid. So already in the first 2 years or 3 years post the pandemic, we managed to produce by design medications that use so much different modes of action that even HIV would envy because of very specific effort. So people sat with a pen and paper and said, okay, we're going to target step A or step B or step C and design a molecule to block it. And that is the beauty of the modern antiviral synthesis. If we find a chance at the end of the talk, we're going to speak about the what do you call it, the method, the lessons that we've learned. Okay, so that's the second bit out. We wanted to speak on the third part, which is the herpes. Before I go, do you want anybody want to comment or mention anything on the previous two parts?
Yeah. Yusri?
Yeah.
Oh, thank you so much. This is like very interesting. I know you made it very simplified and helpful. You want to talk about the nanoparticles? There was an article about like using the nanoparticles to deliver the drugs into the cells. And this is something interesting. Yeah.
Yes, absolutely. So the neutralizing antibodies, the peptides, the small molecules, these are things that you can make into tablets and people can just swallow, or you make them if they've got drug properties into an injection, and then you can infuse it or give it intramuscularly. There are some of these, especially the nucleic acid polymers, and especially the CRISPR-Cas systems, where they're probably going to be working very well, but there's no clear way of how to deliver this. And you remember at the onset of the pandemic, when people were thinking, okay, we've got mRNAs, we can just try to use mRNA vaccines, and people were like, okay, how are we going to deliver it? And if it wasn't for the work that was done in the decade before of finding carriers, finding something that will carry this mRNA into the cell without it being destroyed in the tissues or in the bloodstream immediately after it's injected, we couldn't have been able to produce the mRNA vaccines for COVID. So this product here called EBT-101, can you see that for HIV?
Yes.
This is a CRISPR-Cas9 product that was actually inserted into an adeno-associated virus, like a gene therapy, exactly like what is used in gene therapy, delivered specifically to excise the provirus out of the human DNA to cure somebody from HIV, for example. We're just cutting the HIV out because that's what the Cas9 system works. So you either put it into an adenovirus, like what we've done for the Oxford vaccine, for example, for COVID, or you formulate a nanoparticle to deliver these systems. So the nanoparticle systems is not an antiviral itself or antibiotic itself, but it is rather a vehicle to deliver the load of the antiviral safely into the cytoplasm or into the cell surface. So that's the idea.
So they can deal with latent like infections, right? The reservoir, right?
Oh yeah, absolutely. So of course, if you've got an antiviral that can work in latent reservoirs, then of course that is going to be. I think the nanotechnology is going to be. It's already ahead of the antiviral bit. So, for example, what would stop us from producing more CRISPR-Cas13 or 12 or 9 products now, and use technology that is already being kind of honed with the vaccines. I think what is stopping us is that we don't know much about these systems themselves. And if we deliver them inside, what is going to happen long term? So the safety issue is a huge thing that has not been clarified yet. But the delivery system with the nanoparticles are going to be much easier, I think, in the next few years. Okay, herpes, viruses, and antiviral advances in hematopoietic stem cell transplantation setting. As of course you know, we've got the 8 viruses, 1 and 2 and 3, VZ and 4, EBV and 5, CMV and HHV 6B. I haven't written 6A because 6A really clinical role is dubious. Nobody really believe that it causes a huge problem at all. And then HHV 8. So these are the viruses, and we're going to touch on them very, very quickly, except for CMV. We're just going to take a little bit more time talking about CMV because it will help us with everything else, and because it's the most important pathogen that we deal with on day-to-day basis in the hospital, especially with HIV being now an outpatient kind of specialty, and viral hepatitis as well. CMV is the one that we deal probably with a lot of times on the wards. So human herpes virus type 5, CMV, we know is a giant among viruses in terms of the size and everything, and the implication of acute infection are of really little relevance. However, the interest is in chronification and latency and the development of immune responses that accompanies the host for the rest of their lives. Seroprevalence is very high, but differs really from one place to another with age and with the socioeconomic status. The primary infection is like an infectious mononucleosis-like illness, and the virus reservoir in the body is the CD34 positive and CD14 positive cells and the monocytes, and also it targets the epithelial cells and endothelial cells, so it's everywhere. And the cellular response is crucial to controlling CMV, with CD4 positive and CD8 positive T lymphocytes directed towards the P65 protein. And when the control is well set, then infection becomes indolent. However, a lot of effort is done by the immune system to control the virus. The immune system dedicates 10 to 40% of the CD4 cells in the peripheral blood are actually specific for CMV, and this is just to tell you how much dedication is given to this virus by the immune system. So this is his life cycle. Because we've done the most difficult life cycle, this one is going to be easy, and you just follow the numbers. A virus, a CMV virus, binds to a receptor, fuses with the membrane, and then releases the capsid that contains the nucleic acid into the cytoplasm, and by breaking the capsid, and then the nucleic acid gets transferred across the nuclear membrane like any respectable DNA virus, and then the DNA gets replicated under the effect of the viral DNA polymerase, and then the DNA get packaged, cleaved and packaged, and then encapsulated, and this capsid with the load of nucleic acid inside it get transferred out with what they call egress. And then outside the nucleus, the capsid that egressed will meet the rest of the bits and pieces that we've made in the ribosome, and a new variant will be made, and then will get released out. So these are 9 steps: binding, capsid release, genome release, replication, DNA cleavage and packaging, assembly of the capsid, nuclear egress, assembly again of the virus, and this is the virus whole. Here, this is the capsid, and then release. Now let's talk about the 4 or 5 medications that we use against CMV. So there is a very important step, which is the viral DNA polymerase catalyzed replication. 4 of the antivirals that we use for CMV target this enzyme, the viral DNA polymerase, and it's called UL54. It's the product of the gene UL54. So it is targeted by cidofovir, by ganciclovir, by foscarnet, and by maribavir. The 4 of them target the UL54. Okay, while letermovir targets the viral terminase complex 51, 56, 89. These are just one complex, and normally the mutations within the 56 will confer resistance to letermovir. Maribavir is a little bit different from the others in the way that it has got more than one target. All the others have got one target except maribavir. It targets the polymerase, DNA polymerase. It targets the capsid assembly, and it targets the nuclear egress. And that's why it's very important now to treat resistant and difficult to treat CMV. And it's been a revelation, the recent introduction of maribavir. Okay, ganciclovir, one word about it. Ganciclovir is a pro-drug. It wouldn't work on the UL54 like others if it is not for the viral protein kinase that gives it a phosphorate group. So it phosphorylates it, becoming ganciclovir monophosphate. Then hand over to cellular kinases, and it gives it another 2 groups of phosphate, making it ganciclovir triphosphate. And this is the molecule that can actually act on the viral DNA polymerase, inhibiting it. Cidofovir doesn't need the viral protein kinase. It's made from the factory with its phosphate first group on it already. So when it comes here, the cellular kinases gives it 2 more groups, make it triphosphate, and then it acts on the UL54. Foscarnet, on the other hand, as I said, is a pyrophosphate analog, and it doesn't need phosphorylation by the cell, and it doesn't cause phosphorylation by the virus. It goes straight away and acts on that. So mutations in the UL54 can confer resistance, separately or overlappingly with the ganciclovir or against cidofovir or foscarnet, and some of them confer a bit of resistance to maribavir, but maribavir can overcome that through other means. Okay, so that is the CMV antivirals. Now we're just going to move a little bit more and see how these medications are deployed. How do we use them? Okay, so we know the burden of CMV disease is huge. We know it can affect the brain or the eye or the lungs or the stomach, causing the colitis and the retinitis and everything. But one of the bigger impacts of the virus is on the alteration it impacts on the immune system. Basically, it plays tricks on the immune system, and is implicated in the causation of GVHD, in causing immunosuppression itself, and of course in opening the door for fungal and bacterial infection to wreak havoc and maybe kill the person. If it's not killed by CMV itself, it's by how CMV allows other bad guys to work. And of course, we know CMV is associated with non-relapse mortality in hematopoietic stem cell transplantation. The current management of established CMV disease requires a lot of trade-offs, because if you want to control the virus, you have to maybe improve the immune system by taking your foot off the gas as far as immunosuppressants is concerned, and vice versa. If you take some of the immunosuppressions, then you will lose the the graft that you are actually trying to protect. So it is a difficult situation. And we normally look at CMV disease in terms of risk factors, and risk factors themselves are divided historically into early problems and like first few months, 3 months, first 100 days and beyond the first 100 days. And we know that the general risk factors like seropositivity and advanced age and the type of transplant, the conditioning regimes used, these are very well established risk factors that will apply to first month or second month or even first few years post-transplantation. So I'm not going to go into detail about that, but what I wanted to say here is is that if you just look at the last line, the CMV presentation, in the first month, you may see viremia. So a lot of people will become viremic by day 20 or day 15. But you hardly see any end organ disease in the first month. That changes in the next 2 months or so with CMV pneumonia, the most common target organ disease seen in the first 3 months. Beyond that, reactivation will still be there, and especially in high-risk patients, and the virus will and can still present with any disease, whether pneumonia or colitis or encephalitis. So, what are the major goals of managing CMV with antivirus? First is we want to prevent CMV reactivation, because if we prevent that, organ disease is not going to happen. But if it reactivates, then we would have to get in there by preemptive therapy to stop that reactive or reactivate virus from causing disease. But if the disease already happens despite your best effort, or if your patient comes late, then you would have to treat them, and the treatment preemptively or as treatment for disease are exactly the same. So what has happened in the last 3 decades? A lot of advances happened. So the first one is the improvement in diagnostic tools. The second is the shifting from preventative measures that I've just described, like preemptive, to prophylaxis, just prophylaxis. We don't wait until the virus reactivates and then try to treat it. We just prophylax. And the third bit is the difficult to treat CMV infection. And because we don't have time for all of them, I've taken this one out because this is the Ribavir one, and I haven't got time to show you the data for it, but you can take my word, it's been a revelation. Okay, now, advances in diagnostic tools. This is the first big advance that happened in the last 30 years. And I'm sorry, I mean, you cannot invite a virologist to give a talk and expect him not to take a few minutes talking about lab stuff. So I can't resist this. So we used to do tissue cultures, not long ago. Human embryonic cell lines, fibroblasts, MRC5s, and we inoculate them with blood or with urine or with respiratory samples. And we wait and see sometimes 10 days, sometimes 20 days before we see the damage that is happening in a specific way to these cells called cytopathic effect. And we know that somebody is infected with CMV. And that was very early days. Maybe that is changed in the early 90s, late 80s, when shell vial culture came. So you use the same cells, but you put the inoculum with whatever respiratory or blood or urine or CSF, centrifuge it just to aid infectivity. Next day, you put a monoclonal antibody, and you will see that some cells have been infected by, the highlighted fluorescence in the nuclei of the cells. It is not quantification, but it is just quicker. And then we have the PP65 antigenemia test that was, I think, introduced in '89, but it is very good because it's sensitive, it's quantifiable, type of test. And I think it was in use in some places until 2010, before it was everything's kind of replaced with real-time PCR and viral loads. And even these viral loads were not perfect from the beginning. I remember as a consultant, in Nottingham, in 2007, 2008, when a viral load of 100 copies per mil in our lab. The same sample will be sent to another lab, and the other lab says is 100,000 copies per mil. So there was no standardized way. So the standard that I used for measuring viral load sniff was not great until 2010, 2011, when the WHO introduced a standard, and everybody now is reporting their results as international units per mil. Not 100% done everywhere perfectly, but at least now we can actually make common guidelines. So if I say to you, you use this medication when the viral load is X or Y, and then you can use that guideline in Newcastle or in King Faisal in Riyadh or in New York. So you can use it wherever. And the latest kid in the block is the quantiferon CMV test. It's an IGRA test. It's just like quantiferon TB test. And when used in combination with the viral load, is found to be really useful in stratifying patients at risk of CMV disease. And many centers now use it. It's not widespread, but many units use it. So it will tell you maybe who is maybe got some immunity against CMV. Already now they have recovered their cellular immunity. They can produce interferon and they can fight the CMV. But a negative test does not automatically mean that a clinically significant CMV reactivation is going to occur. It wouldn't. So now we've got these tools. We know exactly what's happening in the body of a person, of a patient, transplant patient, very well, and that helps us to deploy medications, including the latest comer, which is letermovir. Letermovir is excellent, and I'm going to dedicate the next 3 slides for it, but it's got a very low threshold for resistance. So you can use it as prophylaxis, but if there's a breakthrough and the virus goes up, you cannot use it. And thereby, units that use prophylaxis now are more dependent now on follow-up and things on laboratories, given good results or timely results of CMV viral loads and possibly CMV quantiferon results. Okay, so that is the first big advance, diagnostics. The second is the shifting in the preventative strategies. As we said before, we just give them preemptive therapy. And at that time, 80% of CMV seropositive people who receive allogeneic transplants reactivate CMV. Okay, but we monitor viral load first, and we don't give them treatment until they become viremic. Why? Because of cost at that time, a low time, and because these drugs are toxic. The ganciclovir and the ganciclovir, especially in somebody who has a bone marrow transplant, you don't want to kind of jeopardize the way their marrow is working or is going to be working. But in reality, that was a bit of a fallacy, because everybody who get a high risk or an allogeneic transplant and reactivate, we said 80% reactivate, they almost always just keep the virus and may just get worse. So if somebody gets a viral load of 150, the chances that this person will spontaneously control his viremia is 14%. So very low. This is 150. While if they reach 1,000 copies per international per mil, 100% of the people who reach 1,000 never been able to control the virus in there. So they all needed, all these in gray needed post-exposure prophylaxis. Essentially, you're giving post-exposure prophylaxis at some stage to everyone. And worse than that, there was no clear impact on mortality. And I'm going to show you a retrospective study from Fred Hutchinson Center. They just reviewed their pre-prophylaxis era, the preemptive therapy era, and so and just showed what they have been doing. This is 2016, but I think they looked at their results from 2010 to 2015 or something like this. And you can see here, this is a CMV viral load. They used viral load as a time-dependent risk factor for overall mortality and non-relapse mortality. They looked at 926 patients, and in the top panel there, they looked at the overall mortality. And in the bottom panel, they looked at the non-relapse mortality. So the disease did not come back. The cancer is gone, but the patients died. They describe the data in the left-hand side here in A and C using viral load as categories, as categorical values. So less than 500, any positive, 500 to 1,000, more than 1,000. And here they looked at it at threshold levels. And you could see very clearly here, maybe I have to mention first that the overall mortality in this group was 30%. Okay, 30% died. But how many of these 200 and something people who died has their death directly attributed to CMV? 3 out of 263, 1%. So maybe the antivirals were working in preventing CMV related death, but nothing else, because they are all CMV positive. The other thing is, if you look here in the overall mortality, first 60 days, and you look at the viral load here, 250. Any viral load that is more than 250, which is not really huge, have got an adjusted hazard ratio of death of 19.8%. So the conclusion from that paper overall was that the viremia was associated with an increased risk of mortality in the first year, especially the first 60 days, independent of the use of preemptive therapy. Not only that, but also is evidence of positive dose-response relationship. Look here in this bottom graph here. Every time the viral load increases a bit from 250 to 500 to 750, the mortality, the adjusted hazard ratio of death increases. So it's got a dose response relationship. Okay, so we're doing that all this time. We needed something to stop this viremia from happening in the first place, and came along letermovir. So this is the results of the study of the phase 3 placebo-controlled trial of primary prophylaxis of CMV seropositive patients who are recipients through 100 days. And I can say that the trial clearly showed significant reduction in clinically significant CMV by week 24 post transplant. Of course, we've mentioned already that this is the letermovir, that is the terminase inhibitor of CMV. Okay, and I'll show you very quickly also this slide without really describing it in detail, but you could see on the top here is the clinically significant, which is the primary outcome, the efficacy in the population, and according to risk groups. Here, high risk groups, and here, low risk group. So somebody who is a low risk group for CMV, the cumulative risk of infection in him with letermovir is much better and significantly different from somebody who had taken a placebo. So that is, on the basis of these results, the medication was approved by the FDA as for prophylaxis. And the impact of this medication has been further elucidated after the licensure, and that was shown through multiple real life cohort analyses. The bottom one here in blue is a retrospective cohort study published in the Clinical Microbiology and Infection Journal of ECMID, and it just demonstrated the protective effect of letermovir related to lowering of all-cause and non-relapse mortality in seropositive CMV patients. And there is also a systematic review by Fires in 2022, and I'm just going to give you the headlines of the systematic review because it's big enough. It included 48 unique studies, 7,000 patients, and it showed letermovir prophylaxis to be associated statistically significantly with reduction odds in or of reactivation, and the p-values are included there, and this is the pooled odds ratio with clinically significant disease and with clinical disease by day 100 and day 200 post allogeneic transplant, and also with all-cause mortality and non-relapse mortality. So that is being established, clinical trials, cohort studies, and systematic reviews, and now it is the gold standard. Every unit around the place will use letermovir as primary prophylaxis against CMV, which is fine. However, here, this is a recent letter, I think this month from IDSA, and I face this every day on the floor. People are using letermovir against CMV in CAR T cell recipients in the same way they use it for hematopoietic stem cell transplant recipients. This has been proven. I've just showed you the papers and everything, and now established practice, but that is not being replicated actually yet on the CAR T cell recipients. Nevertheless, very expensive, and just people giving it without really much of thought. I use it, but I use it in very specific selected patients of CAR T cell, which I think the risk for reactivation of CMV is maybe similar to hematopoietic stem cell transplantation, especially those who have just failed a transplant. So they had transplant before, but those who never had a transplant before maybe do not need CMV or letermovir as prophylaxis. So that's a thought that I thought I would share with you from my daily practice. Okay, so if somebody's got CMV DNA, viremia, finished. So you need to treat them. You treat them. When do you treat them? We mentioned the threshold earlier, how the diagnostics have enabled us to make better kind of cutoffs, clinical cutoffs, and this change varies from center to center. In MD Anderson Center in Texas, guidelines to use these levels, and we use them here in Riyadh too. Low level or low risk patient, autologous or more than 100 days post transplant. If the viral load is more than 1,000, you start treatment. In moderate risk, you start them if they are more than 500 only, and if they're high risk, like cord or haplo or mismatched or T cell depleted, even if they're just 150. And you remember I showed you in that graph, I said if it goes beyond 50, it's highly unlikely to be kind of spontaneously controlled. It will not resolve, and that's why 150 is being taken as a cutoff for high risk to start antivirus like ganciclovir or biganciclovir or foscarnet, the broader spectrum antivirus. Okay, maribavir, we haven't mentioned it, but this is the agent that we use to mitigate the impact of resistance and refractory CMV infection. Okay, in autologous cells transplant, we don't know much, but it's not a huge deal. But beware of high-dose steroids and irradiation and the use of purine analogs and Campath antibody. That's it. So that is CMV. It took all our time. What shall we do?
15 minutes or 20 minutes after overtime, but maybe you can open the floor for questions, or if you have any more things to add.
Yeah, I think
We
we've
can
done
do another
enough.
session. Yeah, Yusri, if you have still
Yeah, we
like more
can
stuff. This
actually
is very interesting.
I am happy. I'm happy to
Yes.
complete the rest of this one next time, because yeah, I can do that.
Yeah,
I'm happy to do that. The lecture
yeah,
is
Yusri,
there.
this is very helpful. You don't understand. We have so many questions, and I think I don't want you to rush your presentation. So if you still have like all slides, I think we can do another session. What do you
Yeah,
think of this?
absolutely. In fact, as I was thinking, this is not going to happen. Okay, this is not going to happen because it's just difficult to go through them very quickly. But I thought, I thought maybe I need another session, especially dedicated to resistance in
Yes.
non-HIV antivirals. So I think we can just complete the rest of this another time, and then maybe just have a discussion around that issue of resistance.
Sounds good. Yeah, we're open
good.
for next month.
Yeah. So let's discuss what we said already. If you've got any questions.
Yeah, so I can start. Yusri, thank you so much. I mean, I have so many questions actually, but I would like to just start with this letermovir compared to the ganciclovir. I mean, it's very like we're very interested in this drug basically, and I wanted you to talk about the immunosuppressive effect of the ganciclovir. Letermovir does not have the same impact, right, on the
No.
pulmonary
No.
suppression. Okay.
No.
Okay. Okay.
No,
And
it
the main
doesn't.
like.
And
Uh,
yeah. Yeah.
yeah, and the
Go
main
ahead.
issue is the cost now. Is the prohibiting the cost?
Yes. I
Okay.
mean, the main problem with letermovir is the cost. That is the main thing. But it is completely offset
Okay.
by the impact it has on mortality. In
Mm.
fact, now all these retrospective studies and real life studies have shown that the gap between somebody who is allogeneic stem cell transplant recipient who is CMV IgG negative, somebody who's completely seronegative. So his death has nothing to do with the virus at all, okay? And somebody who is seropositive, CMV, who is on letermovir, their numbers are exactly the same. While I showed you earlier that when you use preemptive therapy, there was no impact on survival. So the gap between seropositive and seronegative has been now bridged by letermovir.
Have we started like seeing resistant to UL56 mutation or any?
Letermovir.
Yeah. I mean,
For
has there
letermovir.
been
Oh yeah,
reported like
absolutely.
resistant with mutation in UL56?
Unfortunately, unfortunately, it can become resistant very quickly. That is a problem because and it's not designed to treat somebody who's viremic because it becomes quickly resistant. And that's one of my worries about using it haphazardly. So we use it in transplant. We use it and we monitor our viral load every week or so for that period. And then when they're out of that period, then it can be less. But once we see a breakthrough and the virus just going, 200 or something like this, anything more than 150, we'll have to revert to an antiviral with a higher threshold for resistance, because otherwise we're going to lose it. Not just lose it, we lose it for that specific patient, but it's going to be of no use if the virus can just go through it once it becomes abundant in the bloodstream.
Thank you, Yusri. Thank you so much. Ahmed, do you want to go?
Yeah. Thank you, Yusri, for a nice talk. Just question, Yusri, about the when you have a clinical suspicious of CMV disease, and with low or undetected CMV viremia.
Yes.
When you ask for biopsy, what is
Yes.
what is your cutoff marker when and what to do in that case?
Yeah. So, okay. So there is one phenomenon that is very well described in CMV is called compartmentalization. So the virus, because it's a little bit sophisticated compared to other viruses, it can just behave differently in different compartments of the body. So I've had patients where they've got, confusion and like they were encephalitic. Their viral load in the blood is zero. It's not detectable, the viral load. And I tapped their CSF and they've got 100,000 international units per mil in the CSF. So that is just to illustrate to you how the compartment will work differently. So if you're suspecting CMV disease, especially in somebody who's high risk, who's been on antivirals or not on antivirals, you cannot just rely fully on the viral load. The viral load in the blood will be very useful if you're following them from the beginning when they are negative. Then when they become positive, you give them what do you call preemptive treatment to stop them from having target organ disease. And then also you can use that viral load to see how your antiviral is working. So it's dropping and then you're happy. Or it not, then you think of resistance and you ask for the UL54 or 97 mutation to be checked. But if you see a patient who has been CMV before, positive or high risk, and their viral load is negative or low, that doesn't exclude disease in other places. And the biopsies, specifically the gut biopsy, has been the gold standard. So we need to see the histopathology finding, the histological pathognomonic changes that tell us that there is an active CMV disease there. And that is still the practice here in King Faisal. In Newcastle, we have been using viral load as well to quantify the virus in the cells, and we try to kind of have some control of it by a housekeeping gene to know how many cells were there. And viral loads more than 150 copies per mil were found to be kind of a surrogate marker for someone who is going to have a histological diagnosis if you look for the virus, or if you look through immunohistochemistry or through DNA probes. So we're using the viral load more and more, but this is not standardized across the world. Still, the histology, looking for the active virus in the tissues, is the gold standard still.
Yeah, thank you.
I hope I have answered the question.
Oh, you did, Yusra. I think the highlight is what important. We face a lot of difficulty when we ask for tissue diagnosis.
Yeah,
it's not only on CMV.
It always helps if you've got a friendly histopathologist who's interested in the subject. so they become like experts in it, and then I've got a friend or two who are histopathologists. So when they say to me this is CMV, then I take it. And if they're absent or they're on holidays, I probably sometimes wait and see if somebody can review, because interest and seeing a lot of these things help.
Thank you, Yusri. Yusri, if you can, maybe in the next presentation, if you don't have time for this now, if you can highlight the CMV disease in immunocompetent patient and CMV in the era of biological agents.
In the biological agents.
Yes.
Yes.
Well, Yusri, yes, Ahmed, I was just gonna mention this, Yusri. I have like two patients, like they're immunocompetent, they're viremic, and they have cytopathic effect in pathology, and they're just like driving us like crazy here. I don't know, like, and then Yusri also like one more question, like just to add to Ahmed's. I was told previously by one of the people who trained me that HIV and AIDS, CMV is not a disease in AIDS patient. Is this an
Yeah.
accurate statement or no?
No, of course not. Of course not. I mean, pneumonia maybe is not a problem, but of course, people forget the retinitis in HIV. Of course, when I was writing this week, I was really just focusing on the antivirals rather than the disease. It's just the how to deploy, how to use antivirals. But the question that you mentioned about immunocompetent is, I think, very important. And see things like Guillain-Barré, okay, in immunocompetent people who come with just GBS. In the books, people will say, oh, people have had Campylobacter, people have had something that isn't it. I think the last 10 cases I've seen of Guillain-Barré were all following acute CMV in an immunocompetent person. Proper acute CMV with an IgM and low avidity IgG that matured after 6 weeks, and a viral load. Not always the viral load is very high, but now I think, okay, somebody need to tell these neurologists in their books not to write Campylobacter anymore as the first one. CMV is probably the first one that causes this problem, especially in affluent societies where people grow into adulthood while they're still seronegative for CMV. And we know, seronegative adults, suffer with virus generally. We've seen it in COVID. We know that people, suffer with hepatitis A, with jaundice, and, while children just do not even have it symptomatically. And, varicella, people die in adulthood if they get varicella with pneumonia and everything. But as children, they just run around with their spots. So CMV possibly is doing something similar in many diseases, but it's not very well highlighted. The other thing is that people who are in ICU, who are supposedly immunocompetent, who've got CMV in their blood, we don't know what to do with them. And the last category, which is maybe the most kind of researched, is CMV in the gut of inflammatory bowel disease patients, or people who are not inflammatory bowel disease, but they come with CMV colitis. And through my career, I had at least 2 patients who came with colitis, with bleeding, who needed either resection or embolization or wiring of their bleeders, and they were completely immunocompetent people who came with CMV colitis.
Yeah, that patient I had ended up getting colectomy. He was completely immunocompetent. And as you said, because it can suppress the immune system, at the same time, he was growing MDROs from the blood and urine. And this was kind of like difficult case. But thank you, Yusri. This is very helpful.
It's amazing. الحاجة الثانية that people do not also know is there are things like rare presentations of viruses. So you read in textbooks, somebody says EBV, acute EBV will cause splenic rupture, for example. How many people have seen a splenic rupture? And but it does happen. But in order for the individual physician to see a case, they will have to have looked after 10,000 patients. So it will come one to your clinic, one to my clinic. You are working in the East Coast, I work in the West Coast. People do not see that. But if you're working in a place where all samples are sent like laboratories, and I think that helped me because I was just, seeing all sorts of things because the samples come in with clinical queries and people phoning. So I've seen a 9-year-old girl who died of EBV, for example, encephalitis. She's immunocompetent. She was absolutely fine until a week before in the school. But we proved that she got EBV now is just a rare thing. And I'll go back to the analogy in COVID-19. You will have a very rare event that is associated with something, and you need to have 4 million people who are all followed up with a very good database before you can pick up that side effect, because of that complication. If you don't do it in one database looked after by one group, the Scandinavians wouldn't have known about the clots that happen in the head following the Oxford vaccine. It's just because they have the database open in front of them and they saw that this thing has happened to 3 people or 4 people out of the 4 million they saw. Otherwise, it would have taken maybe a few years in a normal medication that is being sent to pharmacists around the world for people to discover that.
That's interesting. I mean, this EBV, speaking about the EBV, I mean, we all know about this paper with the association with the multiple sclerosis.
Yes.
It's kind of like there's a first proven evidence of infectious agent linked to neurological disease, which the MS, I mean.
Yes, absolutely.
It's fascinating.
Yes.
Yeah.
Thank you, Sri, for the outstanding and comprehensive presentation today. And thank you for all who attended our session today. And we'll see you next month in a new grand round presentation from Sudanese Infectious Disease Association. Thank you. Have a good day.